Ellie, welcome to the show.
Pleasure to be here, Harry.
So, Ellie, I don't even know where to start. I mean, there's a million different things that we could talk about, and but I guess for everybody who's interested in this space, and whether they're young or even thinking about what's happening in this space, like how did you start in this whole area? And really, you're like on the bleeding edge of all this different technology shifts within precision medicine and genomics. So tell me the story.
So yes, it's been a very eventful decade, and very much the radar of at least my career trajectory has been these technological advances, starting from the Human Genome Project to where we are now. So I can tell you a little bit more about it in that I was 18 years old, just about when the Human Genome Project was coming into maturity. So
now you're making me feel old.
Not at all, not at all. And I remember just being entirely fascinated and captivated by the concept that there was this international effort of scientists coming together to decode the sequence of the human genome. And without really having a deep understanding of what that even meant at that time, I knew that that's what I wanted to do for the rest of my life. And I should say that I was a kid in Greece, and I very vividly remember the moment where I was walking down an old street in Athens. It's summer, everyone has their windows open and they're listening to the TV. And I was just tired of listening to the question, what will you do when you grow up? Mm-hmm. And as I was walking down the street, I hear about the announcement of the human genome through a TV channel. And I remember the moment where I stopped and my heart just said, that's what I want to do. And essentially that set off a trajectory where I packed my bags and I went to the UK, to my entire family's despair, to study genetics. And that was the first time where I realized how fast science is, in that my first day at the auditorium of my genetics course, my lecturer comes in and says, the human genome has 80,000 genes. And every year when we started our academic course, that number went from 80,000 to 60,000, and it went all the way down to the 20,000 that it is now.
Yeah.
And I think studying a discipline at the time where that discipline is almost being reborn or reinvented through a big technological advance was one of the most rocket fueling experiences when it comes from a young individual who goes through university and is ready to go head on into identifying their career path.
Well, and I have to tell you, I still feel like it's going through the technological revolution and changing. I always tell people, I'm like, I don't think I can read fast enough to keep up with the changes that are happening from multiple directions. And so whenever anybody makes like this scientific prediction, I'm like, okay, let's see how that goes.
Yeah, 100%. That is one of the most fascinating things about being a scientist, is that you never come out of college, you never graduate. You will always have way more to learn and understand than what you have mastered in terms of both technology and facts.
I wish we had scientists that thought like that more than you. I remember when someone said, dark matter is not important in the genome. I was like, how can you say that? We don't even know.
And then came the ENCODE project.
So tell me what you're doing now.
So yes, I didn't answer your first question. So where I am now is I'm an assistant professor at computational oncology at MSK-CC. That's Memorial Sloan Kettering Cancer Center in New York. And I really feel that I have the privilege to work alongside some of the best scientists and physician scientists in oncology to very much understand how we can use both genomic technologies as well as genomic data to inform patient care.
Now, I know that you've worked on like different technologies on the way. I mean, you seem like you're constantly graduating to the next level. Where did you start? I mean, I know that I think it had something to do with colorectal cancer and microarrays. Was that correct?
Correct. So as I was graduating from my university, I had to choose where I would go on and do my PhD. And I remember I interviewed through so many labs, and that's so important in science, in that you really need to follow something that makes your heart tick. And for me, that has always been at the intersection of new technologies and data. So that brought me for my PhD at the Institute of Cancer Research, where to do a PhD, I was one of the first adopters of microarray technologies at that time, to do genome-wide linkage studies. So that is to interrogate the genome of families where there are multiple occurrences of colorectal cancer and map 10,000 points across the human genome in order to identify chromosomal regions that may associate more often than you would expect by chance, right, or segregate patients with cancer. Then these led me to one of the first genome-wide association studies where we used population genomic approaches to identify common genomic regions that predispose to leukemia. And at that time, there was the big switch from high throughput genotyping platforms such as microarrays to next generation sequencing technologies, where now for the first time, we were in a position to sequence the entire exome, all the genes in the human genome, or the entire genome. And that took me straight to the Wellcome Trust Sanger Center, one of the best genome centers in the world, where I had the privilege to be part of the International Cancer Genome Consortium. Mm-hmm. And that effort was very much concentrated in performing whole exome or whole genome sequencing in the 50 most common cancer types, with the aim to identify all the genes that are mutated in cancer. And just to give some perspective on that, this initiative started only 12 years ago. Yeah. And it was only 5 years ago where we could say that with confidence we had concluded the first catalog of the genes that are mutated in cancer. So the next question that came from that is, what do we do with all this information?
Right. Well, it's funny. I mean, I don't know if you saw, there was somebody that wrote a piece the other day, again, one of these pieces of, there was this promise of genomic medicine and on what doing the genome would do, and it's done almost nothing. And it drives me bananas when I see these things. I'm like, wait a minute, it's the fundamental foundation for any modern biotech like that you could possibly think of.
But you know what, we're all a little bit culprits for that, because, and I'll tell you quickly my thoughts about that. Every time we discovered a new gene, right, I can't describe the moment of that discovery. And then both the scientific and clinical community, we would come out. I was part of many of those discoveries, and we said we discovered this new gene. And then both with the press announcements, both the publications as well as the media wanted us to wrap up to say, and this is going to help us cure that type of cancer.
Right.
So that led to this expectation of precision medicine, and in a way resulted in not unreasonable hopes, but potentially the timeframes or the steps that we need to go from now to the realization of this vision of precision medicine potentially were not clearly understood. So that's what we're working on right now.
So, I try to make sure that we are talking, people understand like data on the show, like what is the purpose of this data, and So where do you see the different uses in the lab that you're in now, and what is your vision for where you'd like to see it go? Because I know you're very passionate about the direction of this.
Right. So we're now sitting at probably at the middle of the hill. We understand what the building blocks of cancer are, and we have this vision about precision medicine. And precision medicine should be reflected on the entire journey of a cancer patient. So we should start by understanding which individuals are at risk of developing cancer in the first place before they even develop it. Once early symptoms or early screening approaches, we can diagnose patients with early stage disease using appropriate technologies in order to mitigate, so survival of patients or treatment outcomes of patients with early stage disease are much better than the ones with later stage disease. But fundamentally, once a patient has been diagnosed with cancer, we have this vision where they're sitting in their oncologist room next to a computer, and as part of the medical record for that patient, we have data-driven models that consider the age of the patient, the gender of their patient, their genomic parameters, their lifestyle parameters, and they can inform their pathology or radiographic data, and they build an accurate picture of that patient's diagnosis. And also they deliver predictions or proposals with what the best line of treatment might be for that particular patient, given both their genetic makeup, their age, as well as all their other clinical presentation parameters.
So you're talking about you're going to have to use some form of AI and machine learning to take the base data and make it digestible, right? Because even some of the best, that I know have to pour into this data at an unbelievable level of focus and understanding to then, come up with, okay, here's what we think.
Mm-hmm. Exactly. So there is so much hope and also expectation on the use of AI-based approaches in healthcare overall and cancer in particular. So, but for that, and the basis of that is that we can learn from many patients in order to inform the treatment of a single patient. That's the fundamental premise of that. So how can we leverage all the data, historic and existing, as well as prospective data from thousands and thousands of patients, and convert them to confident and evidence-based decisions for the N of one that is sitting in that room with that oncologist that day? And most importantly, how can we empower that oncologist to make the right decision for that very patient?
So, speaking of leukemia, if I'm not mistaken, right, that was one of the fundamental papers that you presented, and you've built a consortium around it to do more work in that area. So hypothetically speaking, God forbid you're a leukemia patient, right? But what if you were telling a patient what steps to take for taking advantage of this? What would you sort of tell them?
So I think we're addressing more the oncologist and the treating groups in order to support and help that patient. And I think the examples that we see in leukemia are also translatable across cancer types. But to give you an example of how unique every patient is, leukemia is probably one of the simplest cancers from a genomic perspective. So just the basic building blocks of cancer is that every cancer occurs as a result of multiple mutations. Usually those mutations happening in sequence, we have a first hit, those cells expand, first mutation, and then we need secondary and third events that eventually will transform a normal cell into a mutated cancer cell population. So we performed a study, and so currently what's happening clinically is that a new patient will get diagnosed, and a biopsy from their tumor or their leukemia is going to be taken, and it's going to be evaluated across a number of genetic markers. And the reason we do that is that so that we can inform the diagnosis of each patient better. We can determine a more accurate molecular subtype and inform treatments for that patient. So we did this exercise recently. We're just wrapping up this study in 3,400 patients with one particular type of leukemia. So that is a single disease that's uniformly treated in the same way by hematologists around the world. Mm-hmm. So in these 3,400 patients, we determined 2,500 different genetic profiles. So that very much showcases why cancer still is today, only 5 years after the completion of the Genome project, a complex disease to tackle with regards to precision medicine. However, using a kind of machine learning based approaches, we were able to reduce this complexity to 15 common themes. So 15 classes, if you will, of genetic themes that not only helped us understand that these represent distinct biological entities, but clinically, patients with each one of those 15 groups present differently. Some of them may be young, some of them may be older, and most importantly, respond to standard of care or novel interventional therapies in different ways.
Right, right. So, it sounds like whack-a-mole, like you're going to need more than one approach, potentially more than one therapy on the same patient, but it really says this standard of care.
At the moment, our standard of care represents more brute force, and we're treating more or less diseases uniformly. Now, through the ability to look at each patient's genomes, we understand that there's a lot of complexity, but then if we study large enough patient samples or cohorts that are where we have matched genetic information with very good clinical annotation and outcomes, we can now bring the AI component into the process and start developing both classification and prediction tools that will effectively become or inform that web portal tool of the future where an oncologist sits with a patient.
Right, and I can see it where if you can bin these groups together, it will send patients down a different path. But not only that, if you're looking at this disease at different stages and you can build a movie, you can dial forward or dial backwards to be able to look at the disease, which can inform drug discovery, patient management, all sorts of information.
So you just touched upon two really important concepts in cancer care. So the first one is that we tend to look at single biomarkers, but we're learning that no patient is a single biomarker. It's not every patient is a complex and dynamic system. It's almost as if you imagine that if you have a chair with 4 broken legs and you try and fix the one leg, you can't expect that chair to maintain equilibrium and be fixed with the one fixed leg, okay?
Elli, you're expecting everybody now to understand complex concepts.
Right. So the question is, how do we understand everything that's broken and try and fix, kind of bring back stability by looking at the whole picture?
Which is impossible to do without AI and machine learning as a tool to assist.
Correct. Or we can get there faster if we bring systematic approaches and data-driven approaches to inform those decisions. So far, we have a number of success stories where we have data-driven models to inform patient care. But these ones so far, and as it should be, are grassroots scientific movements. So how can now learn from those grassroots scientific successes and understand the process, and how can we embed data-driven and AI approaches into essentially the development of infrastructures and at an institutional and national level that can enable and accelerate AI-based solutions in healthcare and medicine.
So now help me here, because what you're talking about is a serious interdisciplinary team of people that need to come together, that have expertise. Because if you could mold all that into one person, these are, you know, yeah, I always think of Star Trek when we get to that type of person, right? But
yeah, it will.
Your team has got to be composed of that.
That is correct. This cannot be materialized by one flavor of a scientist. Scientists today can be treating physicians, they can be biologists, they can be computational genomicists, or data scientists who have trained in machine learning and AI-driven analytical approaches. And my team is very much a reflection of this type of composite.
But that doesn't, I mean, let's face it, you can't have that everywhere all the time, right? This is a, you know, you can't have the winning MBA team everywhere all the time. And so how do you envision this being widely available?
You can think about this in creating the right resource and ecosystem to support these initiatives. And these type of initiatives can be created and supported within academic institutions, let's say, for example, Memorial Sloan Kettering, or through national initiatives. And one can imagine, and we know that, for example, scientists to date have been supported to perform their experiments by accessing biological specimen biobanks. Yep. Every time a scientist wanted to do an investigation or an experiment in the lab, they would go to the biobank and identify some samples and perform their experiments. So we can start thinking about a similar concept, but with digital biobanks. So what about if at an institutional, and why not national level, we start introducing the concept of digital biobanks, where we have infrastructures that allow us to have high dimensional data, genomic, imaging, radiographic, in a structured format that is readily computable, but most importantly linked to clinical annotation and outcome metadata. So once one builds, and these are things that a hospital or a research institute or even a diagnostic company could start thinking about building with partnerships, or this could be mandated through national funding initiatives and promoted, where we now start to build architectures of these data sets. And then this can represent the ecosystem that scientists can access with specific questions to start building models for those patients. We have the option as a community to lead these into a siloed grassroots movement, or we really sit at a point in time where we understand what needs to be done, and we start thinking a little bit more globally and more ambitiously in some ways.
Yeah, no, I wish we were thinking about this at a national level. Like, you know, that would be my, right, you know, if I would be like, if you're getting public dollars, we'd like to see it in this format. But Jesus, just trying to set that up. And then when I think about the IT infrastructure of a normal hospital, I break out into hives most of the time, right? It's not exactly what you would consider the epitome of that technological solution.
The EMRs are challenging, and this is not to say that I don't appreciate the challenges. Like solving the EMR data problem is for a reason one of the most challenging problems to crack around the world and throughout time.
But that requires the EMR companies to want to fix the system, right? And I just, in my heart, I don't believe that is a goal of those organizations, which is why I believe most of the time the change is going to come from externally into healthcare, or places like I think it's Northwell in New York has announced that they're going to build their own EMR because they hate the commercial EMRs.
I think we could devote an entire podcast
yes.
EMR
yes.
challenge. But I should say that coming from, for example, MSKCC has developed a molecular profiling assay called IMPACT, and now the majority of patients with advanced disease that are being cared for at MSKCC have access to molecular profiling of that tumor. Because this is happening at such high throughput for a large proportion of the patients, there's been an enormous effort and initiative to have all that data essentially databased and updated on a nightly basis and available through the CBio portal at MSKCC. So these high throughput technologies are now providing the embedding of these high throughput technologies, and the way that that data is structured and linked and visualized through web portals is starting to bring the concept into within institutional walls. And once you have those data sets present, one starts thinking, and certainly the chief of computational oncology, Dr. Shah at MSK, is building such initiatives where one says, now we have that in place, what about we start layering this with additional data?
Yes.
And then you can start buying tumor types or cross-sectional projects. And then as we layer that data, you see that an ecosystem is born that will allow and will accelerate the development of AI-based research in healthcare.
But take one step back. I mean, just having some sort of a system like that, I can see how the payers, when they have some level of standardization of analytics, might be more apt to want to pay for sequencing as a service, right? Because right now I can't say they're jumping up and down, because depending on where you get it, you might get different results or different information. And so a system like this might actually help provide a layer of standardization on top of that.
Mm-hmm. And also for a return for investment, in that we can enter our data into this type of infrastructures, but then the return of investment to the many N of ones of patients that are being treated and cared of by systems that are informed and supported by this type of infrastructures is very significant.
Do you have any success stories that you feel comfortable sharing?
So there are, so this is again a very, with what particular, like let's drill this down.
Where you've utilized this system that has been available or that you're building that has informed, let's say, a unique or insightful therapeutic direction that might not have been chosen from the quote standard of care.
Right, so we've been leading an initiative at MSKCC to evaluate both the feasibility as well as the advantage of looking at whole genome sequencing in upfront clinical care. So as I mentioned earlier, the majority of molecular diagnostics in cancer are looking at very specific genetic regions. They will look at a number of genes. This is a very practical and robust, scalable solution for the many, many thousands of patients. However, in the time that we've implemented these targeting gene sequencing tests, technological advances have happened that allow us to equally rapidly and as cost effectively look at 100% of the genome. So the reason that whole genome sequencing has not been tested or implemented in clinical care yet is because A, it has been very expensive, but most importantly, the scale of the data that generates and the ability to analyze and interpret that data has been put into question, as well as whether this adds any value in the clinical setting above and beyond the few hundred genes we understand very well. So we decided to put this into a test. So we've developed a rapid turnaround, clinical grade, research but clinical prototype whole genome sequencing platform. And we've developed a study for pediatric patients with rare cancers. And we asked whether applying such comprehensive genomic profiling approaches could help us optimize care for those patients. And we're just about concluding that 1-year pilot study. We showed that by working with advanced software engineers, we can turn around whole genome sequencing data from raw sequencing to a report within 2 days. That is within a research setting, but it shows a path of how this might be implemented within a clinical diagnostic laboratory. And most importantly, what this allowed us to evaluate is the sheer impact of now looking at 100% of the data in a cancer genome, as opposed to a few spots around it.
Yes.
And I've been part of these molecular tumor boards, and I've been leading the genomic part of this analysis together with my team. And week on week, witnessing the value of having the complete picture as opposed to a few regions helps us understand a lot more about whether that patient, for example, has an underlying germline defect or a risk predisposition mutation in cancer through kind of a defect in DNA damage response mechanisms. It helps us to map out all the genes or mutations that have gone array in that patient's tumor. It helps us assess the proportion of the genome that has been altered or affected. There are things we call now, such as mutation signatures or mutation processes that may allude towards potential therapies, as well as evaluate if more systemic, not only targeted, but also systemic therapeutic approaches, such as immunotherapeutic approaches, would be suitable for those patients.
Amazing. Having the whole picture actually gives you better information. What an amazing concept. I mean, this is like the blind man who is feeling the trunk of the elephant and trying to figure out the rest of the animal, right? It fascinates me that, this is such a novel concept, right? I mean, you're laughing, but I've had to deal with people in the industry for a long time on these different issues.
Today, you speak to most oncologists and they will say, we have to also realize that oncologists are looking at those reports and they say, how do I translate this into an actionable clinical intervention? So the thought of looking at the entire genome can be first inundating, but second, we haven't done that yet in clinical practice. So
But I would argue with you, I would argue that there are a lot of oncologists that don't even look at the report or don't even order the report to begin with.
Mm.
Then there are other ones that look at the report, the data is there, and they decide that standard of care or some other pathway is a better way to go and not follow the data, right? So,
I think
you know,
it's down to us. I think it's in our hands for both. We're bringing new technologies and new concepts. And I think the responsibility is in our hands to work alongside the oncologists and the clinical community and identify and surface those examples that are really life-changing. In our pilot, we had patients at their end of treatment where whole genome sequencing allowed us to identify targets and drugs that are available today that we would have never thought about giving prior to now. And they were curative. But we need to move from anecdotal examples to demonstrating and providing the evidence of where and when that adds value, and a clear path forward for the implementation, the safe implementation, and most importantly, without adding burden to the oncologist, but embedding it within their comfort zone. And once we start materializing change and showcasing the value that we can bring, we will never go back. Because I know that
I'm with you. I mean, I want to see that. I always have trouble sometimes when I'm talking to other people because they sort of fight it or they argue with it. I mean, I remember there was the first time I gave my talk about genomic medicine, and somebody asked me to come and give ground round someplace, and I had gone there. And after the talk, this doctor came and started yelling at me. "We will never practice medicine this way." I was like, "Why are you yelling at me? I'm reporting to you the data. It's not my opinion," right? So now, all of that said, you're now at the end of this pilot. What's next?
Well, we need to bring it into a validated clinical test. We need to make it accessible to more than the few and demonstrate at scale the value that more than systematic sequencing approaches can bring to both patient care and oncology care.
But now with a platform like this, I mean, this is a hypothesis, you know what it does, and I haven't seen it, but you could identify diagnostic biomarkers, prognostic biomarkers. I mean, people could be looking at this and utilizing this in a number of different ways that I think we haven't even touched upon.
Correct. And I think these are parallel journeys. So the development of, or showcasing that we can now reliably look at 100% of a patient genomes and enable optimal diagnosis, care, and disease surveillance is a major milestone that we can accomplish. In parallel, there are waves of research that are focusing on how can we first characterize individual biomarkers, right? How can we understand different mutations in genes? What is dosage of mutations? What is lineage of where these mutations happen in terms of which cell type? How do the co-occurring mutations or mutation signatures add information? And then third, how do we understand the clinical relationships of the individual biomarker and then all the biomarkers together as a whole, and how do we develop models or tools that can help us translate each biomarker and composites of biomarkers into actions. And then comes the platform that can detect all of those biomarkers. Okay, so there are two different paths. Does this make sense?
Yes, no, I understand.
So there is what we were talking at the first part of the talk is how do we make sense? How can we incorporate genomic information, and how can we use AI-based tools to inform clinical practice? And there we talked about the development of data-based and evidence-based models that are learning from many patients to inform the interpretation into the N of 1. And the second part is how do we make sure that we capture all the biomarkers in that tumor? And that's where whole genome sequencing comes from. So we as a community, we have this, at least these two tasks to work on. We need to inform both and develop the models that will allow for the interpretation. And we also need to develop the platforms that can rapidly and reliably extract all the relevant biomarkers for a particular patient and inform clinical decisions.
And you know where I'm struggling with this is how to make this standard of care, right? How to roll this out in a way that it's universal, right? How do you have I don't want to call it a call center, but a place where a pathologist can sort of utilize the software and then, right? I'm not trying to belittle it, like, you know, but to get this to the level that we're talking about, there has to be an industrialization, not art, and each place paints its own picture.
Right, or there need to be specific standards. So
you're more hopeful than I am on the standards thing.
Well, the regulatory and compliance system does dictate for certain standards, and we've seen the rolling out of this type of platforms. So in being realistic, there will be implementations of this type of approaches within the big institutes and the big cancer care hospitals. And then there will be in the US centralized service providers from a commercial standpoint. And we've seen many such providers in the molecular diagnostic space in the US. Or if we look across the pond in Europe, you will have government-led centralized laboratories, such as what we're seeing in the UK. So the necessities for some centralization and some standardization in order to be able to apply this at scale. And in order to be able to gather that data that will allow continuous interrogation and optimization of both the technology, the models, and the tools that will inform what will end up in that patient report.
Yeah, I mean, I've been advocating to try to build a genome center in Armenia, right? And to do whole genome and then use, you know, analyze at that level, right? So I can see how if you had a central place to do this with access to software like this,
yeah,
it might make the whole process a lot smoother and easier.
It's going to be a game changer. And countries such as England, Holland, Australia, Ukraine, many countries around the world are starting to build these initiatives. So
maybe that becomes the first place where you license this technology or make it available to them.
We can certainly help with that, for sure.
Well, you know, what else can you tell us about where you think, you know, this genomic approach would have an impact? Where do you, I know it's cancer you've been spending all your time on, but if you let your imagination run wild, where do you see the sort of the next opportunity beyond cancer?
Beyond cancer. So since the sequencing of the human genome, every day we're witnessing the changes that having access to genome profiling technology can bring in healthcare. I would say that some of the biggest and most material changes have happened in the setting of neonates, where we have newborns who will present with a range of symptoms that could allude to a number of potential genetic disorders. And there are programs now in place that will perform whole genome sequencing of a newborn at the first indication that something is going wrong, and then use machine learning algorithms to both identify and prioritize, most importantly, the most likely causal variants that may inform a particular clinical diagnosis. And this leverages from whole genome sequencing data, the incorporation of data mining of the entire literature of symptoms, linking them to the genetic variants, and then coming up with proposed solutions or treatment solutions. And this has revolutionized both the diagnosis and treatment of developmental disorders at a very early and very critical stage.
Yeah, I've had previous podcasts where I've interviewed, the CEO of Face2Gene, and then Dr. Robert Green from up here at Harvard, who's sort of trying to pioneer a lot of the work that's going in this area. So it's fascinating, the impact. I just wish that we would write about these things in the popular press more, so that the average person understood the impact of these capabilities and technologies.
So we've seen some of the best examples of success where we have what we call monogenic disorders, okay? Where there is one more or less root cause of a particular clinical phenotype. And we now try to translate these to more complex diseases, cardiovascular disease, autism, cancer, to name some, or mental health disorders. These are more complex systems. And the fact that it's taking us some time to get there should not be confused with the fact that we won't get there.
No, absolutely.
So it's just that because these are complex systems, it means that we need to understand many pieces of the puzzle. And in order for us to be successful in doing so, we need to pay equal focus on the single feature, and as well as how all the features that we're evaluating as data scientists come together to both result in that particular disease phenotype and how best to manage it. So it's a complex task.
No, of course. I mean, I think about it, and I always say to people, because people want to find the simple answer. And sometimes the simple answer is buried in a lot of different data that's coming together. And unfortunately, the human brain was never designed to look at that many different data points coming together and be able to distinguish one. This is why we have the scientific method. I've looked at a few things, I have a hypothesis, I'm going to go chase it down. But at some point, these complex diseases have so many different things coming together, you can't chase each one down one by one. It would take you a lifetime.
Exactly. So now we have, when it comes from diseases that we suspect have or hypothesize that they have a genetic etiology, we now have a toolkit in our hands that can scan a large space of that genomic footprint, okay? And this is where we can bring, we now start to understand what the roadmap is. So we know what the vision is. We start to understand what we need to get there. And to get there, we need to work in multidisciplinary teams that understand each one of the facets, both disease biology, genetic presentation, clinical care, as well as data management and data analysis. And then we need to develop this roadmap. And this is why, and the roadmap will take either 50 years or it can take 10 years. And the only way to accelerate this is by putting a design phasing process, a design for the architectures and the teams that we need to bring together in order to push this forward. Which is why
I'm giving you a challenge, 3 to 5. 3 to 5, not 10, 3 to 5. I'm getting older. I need this to happen much faster. I think you need to sit down with Francis Collins and work on getting some funding here to move this faster.
We need to have ambitious vision. We need to be ambitious, and we need to be realistic at the same time, but without letting the realism temper our ambition.
We need an Elon Musk in this space. Maybe you're that person, right? Somebody who's going to have audacious ideas and say, I'm going to not just launch a rocket, but land it on a target, floating in the ocean, right? And do what NASA couldn't do.
We need to start thinking like that, 100%.
I don't think that's a problem. I think there's a few people out there that can help with this. Elli, I want to thank you. It was wonderful speaking to you. I'm sure our listeners will have 1,000 more questions. So don't be surprised if you get inundated with emails and calls. But it was great to talk to you. I'm sure I think when we started this, I said we could do a multi-part series on this discussion.
Yeah, it was a real pleasure starting this conversation. I just feel that it doesn't do it, we can't do it justice, but at least starting the conversation is often the biggest and most important part.
Yeah, I mean, I try to convince people, that having a genomic view of your disease is critical. And it's difficult because the layperson doesn't understand that it's at least a map. It's at least a piece of information that you can't get in other ways. And it's critical to how you might be managed in your disease state. So on that note, thank you.
Thank you so much, Harry.